Welcome back. We have a question that reads, a compound microscope consists of an objective lens of focal length 5.0 centimeters and eyepiece lens of focal length 10.0 centimeters placed 25.0 centimeters apart. The final image is formed at infinity. How far is the object from the objective lens? Before answering this question, it is important to know the compound microscope. So we are going to use the ray diagram to explain how a compound microscope works. For this diagram, we have the compound microscope at normal adjustment. And when we talk about normal adjustment, we mean that the image is at infinity. There can be also another adjustment for a compound microscope that is known as near vision adjustment. Basically, when we talk about normal adjustment, like I said, the image is formed at infinity. The eye is maximally relaxed because we are viewing a distant object, okay? But when you have a near vision adjustment, that is when the object is very close to the eye, in order to focus the image on the retina, the eye muscles have to be contracted, okay, changing the shape of the lens. So there is actually a strain on the eye, and that is called accommodation. So when we are focusing near objects, that is called near vision adjustment. But when we are focusing somehow distant objects where the image is formed at infinity, that is called normal adjustment. In our next video, we will be talking about near vision adjustment. This is the ray diagram for a compound microscope that is at normal adjustment. So let's talk about the structures that we have in a compound microscope. As you can see, it consists of two convex lenses. One of them is called the objective lens because the object is closer to it. And the other is called the eyepiece lens. That is the lens through which you look or observe the object. So in short, the lens that is closer to the object is called the objective lens. The lens that is closer to the eye of the observer is called the eyepiece lens. Both of them are convex lenses. So let's talk about how the compound microscope works. As you can see, we have an object here. The rays coming from the object will pass through the objective lens. And as they pass through the objective lens is going to focus the rays to a point here where an intermediate image is formed. This intermediate image is inverted. As you can see, it is real and it is magnified. For the case of compound microscope at normal adjustment, this intermediate image has to form at the focal point of the eyepiece, okay? It has to form at the focal point of the eyepiece lens so that the final image will be formed at infinity. So the intermediate image is now going to serve as the object for the eyepiece lens. What is the function of the eyepiece lens? The function of the eyepiece lens is just a simple magnifier, which means what it does to this intermediate image is just to magnify it. when it magnifies it, the final image is a virtual image that is formed at infinity. We've said that it is just a magnifier, so it means the final image that is at infinity is inverted, magnified, and a virtual image. We have some symbols here. UO is the object distance from the objective lens. VO is the first image or the intermediate image distance from the objective lens. Okay? The distance of the first image, which is also called the intermediate image, from the objective lens is what we call V0 or VO. The distance between the intermediate image and the eyepiece lens is what we call UE. Okay? Remember, the intermediate image is going to serve as the object for the eyepiece lens. And what will the eyepiece lens do to the intermediate image? It is only going to magnify it. Okay? It's going to make it large. So when we talk about the length of the microscope or the tube length, we are referring to the distance between the objective lens and the eyepiece lens. We can say that the length of the microscope is equal to the image distance from the objective lens plus the object distance from the eyepiece lens. All right, this formula is very important. But we have said that for normal adjustment, in order for the image to be formed at infinity, the intermediate image, which is serving as the object for the eyepiece lens, has to be formed at the focal point of the eyepiece lens. So what does that mean? If the image is at infinity, it means that the object distance from the eyepiece lens, that is the intermediate image, is what we call the object here. It has to be equal to the focal length of the eyepiece lens. So we can also say that the length of the microscope is equal to V0 plus FE. All right, since UE is equal to FE under normal adjustment. To recap, the rays coming from the object will pass through the objective lens, and the objective lens is going to focus these rays to a point where an intermediate image is formed. So this is the first image. The eyepiece lens is now going to use this intermediate image as an object and magnify it, forming the image at infinity. So this is how a compound microscope works. Let us write down the nature of the intermediate image. We said that it is inverted. As you can see, if you compare it with the object, it is clear that it is inverted. We said that it is a real image. Okay, the intermediate image is a real image, and also it is magnified. And for the case of normal adjustment, the image is at the focal point of the eyepiece lens. So that is the nature of the intermediate image at normal adjustment. So what does the eyepiece lens do? It is going to magnify this intermediate image so that the final image is formed at infinity. So what is the nature of the final image? Remember, the eyepiece lens only magnifies it. So it means it does not invert it. So it means the final image will be inverted because it has not changed its orientation. It is a virtual image. It is magnified. And it is formed at infinity. Also, when we are dealing with compound microscope, it is important to know that the magnification of the objective lens is equal to the distance of the image from the objective lens divided by the distance of the object from the objective lens. The magnification of the eyepiece lens is equal to the distance of the image from the eyepiece lens divided by the distance of the object from the eyepiece lens. I also want you to know that in some textbooks, the distance of the image from the eyepiece lens, that is VE, is also the same as D. In some textbooks, they will just write D instead of VE. So what is the overall magnification? The overall magnification of the compound microscope is equal to M0 times ME. That is V0 divided by U0 multiplied by VE divided by UE. If you want, you can say D over UE. So that is V0 divided by U0 multiplied by D all over UE. Okay, so that is the overall magnification of a compound microscope. It is also important that you remember that 1 all over f0, that is the focal length of the objective lens, is equal to 1 all over u0, that is the object distance from the objective lens, plus 1 all over v0, that is the image distance from the objective lens, which means that is the distance of the intermediate image from the objective lens. And also, it is important to know that 1 all over FE is equal to 1 all over UE plus 1 all over VE, or we can call it just D. Okay, where FE is the focal length of the eyepiece lens, UE is the object distance from the eyepiece lens, which means it is the distance of the intermediate image from the eyepiece lens, and D is the image distance from the eyepiece lens, which is at infinity when the microscope is at normal adjustment. A quick note: the focal length of the objective lens is positive. We are using real is positive sign convention. The object distance from the objective lens is also positive. The image distance from the objective lens, that is the first or the intermediate image, is also positive since it is a real image. The focal length of the eyepiece lens is positive because it is a convex lens. The object distance from the eyepiece lens, which is the same as the distance of the intermediate image from the eyepiece lens, is positive. Okay, so since the final image that is formed is a virtual image, it means that the image distance from the eyepiece lens is negative. It is important to know the sign that you have to allocate for these parameters. So if this concept is clear, we can go back and answer the question. In the question, it has stated that the objective lens has a focal length of 5 centimeters. That is, the focal length of the objective lens is 5.0 centimeters. The eyepiece lens has a focal length of 10 centimeters. So that is FE is equal to 10.0 centimeters. And these two lenses are 25 centimeters apart. So it means that is the length of the microscope or the tube length. So we can say L is equal to 25.0 centimeters. That is, when we refer ourselves back to the ray diagram, it has stated that the two lenses are 25 centimeters apart. So that is the distance between the objective lens and the eyepiece lens. The formula for that we have stated that is V0 plus UE or V0 plus FE if at all we are talking about normal adjustment. Because under normal adjustment, the object distance from the eyepiece lens is equal to the focal length of the eyepiece lens. That is this. So using L is equal to V0 plus FE, we can determine V0. That is the distance of the image from the objective lens. Note, L is equal to V0 plus FE. That is, the length of the microscope is equal to the distance of the image from the objective lens plus the focal length of the eyepiece lens. This is true when the microscope is at normal adjustment, where the FE is equal to UE or vice versa. So the length of the microscope is 25.0 centimeters. This is equal to V0 plus the focal length of the eyepiece lens is 10.0 centimeters. Collecting like terms, we'll have 25.0 minus 10.0 is equal to V0. So V0 is equal to 15.0 centimeters. So what is the question asking us to find? The question is asking us how far is the object from the objective lens. What is that? That is U0, the distance of the object from the objective lens. So we are going to use 1 all over F0 is equal to 1 all over U0 plus 1 all over V0 to be able to find U0. That is 1 over the focal length of the objective lens is equal to 1 all over the object distance from the objective lens plus 1 all over the image distance from the objective lens. So that is 1 all over the focal length of the objective lens is 5.0 centimeters. Let's say 5. This is equal to 1 all over the object distance from the objective lens plus 1 all over the image distance from the objective lens is 15. Collecting like terms, we'll have 1 all over 5 minus 1 all over 15 is equal to 1 all over U0. 1 all over 5 minus 1 all over 15 is 2 all over 15. This is equal to 1 all over U0. So if we cross multiply, we'll have 2 times U0 is equal to 1 times 15. So that is 2U0 is equal to 15. To find U0, we divide both sides by 2. So this will cancel this, meaning that the object distance from the objective lens is 7.5 centimeters, and that answers the question.